Good afternoon and welcome all. Today we are going to discuss about cytoplasmic membrane. Introduction. The bacterium, despite its simplicity, it contains a well-developed cell structure which is responsible for some of its unique biological structures and pathogenicity. Many structural features of the bacteria are unique to bacteria and are not found among archaea and eukaryotes. The cell structure of the bacteria has been well studied and it reveals many biochemical principles that have been subsequently applied to other organisms also. Now coming to the cytoplasmic membrane. It is also called cell membrane or plasma membrane and it is about 7 nanometers thick and it lies internal to the cell wall which encloses the cytoplasm of the bacterium. Next is structure and composition. Like all biological membranes in nature, the bacterial cytoplasmic membrane is composed of phospholipids and protein molecules. In electron micrographs, it appears as two dark bands which are separated by a light band. So in between these two dark bands are light bands, and it is actually a fluid phospholipid bilayer embedded with proteins. What? The cytoplasmic membrane. It is a fluid phospholipid bilayer which is embedded with proteins. And all of the bacteria possess cytoplasmic membrane with the exception of certain bacteria, that is mycoplasmas. The only bacterium that lacks a cell wall. The prokaryotic membranes lack sterols. So they lack sterols. Many bacterium, however, do contain sterol-like molecules called hopanoids. Like the sterol found in the eukaryotic cell membrane, these are similar to the sterols which are found in the eukaryotic cell membranes. The hopanoids most likely stabilize the bacterial cytoplasmic membrane. So the bacterial cytoplasmic membrane, it is stabilized by a sterol-like molecule called hopanoids. And this molecule, it is absent in mycoplasmas. This is the diagram of cytoplasmic membrane. Here we can see the green color, it is the protein. And this layer is inner layer. And these are the proteins which are core-forming protein. And this portion, this is inner layer and this is outer layer. And that outer layer is possess water-soluble glycerol and phosphate portion. And the inner layer possess water-insoluble fatty acid portion. And this is phospholipid molecule. Because of the two layer, it is also called phospholipid bilayer. This is the another diagram of phospholipid bilayer. This two phospholipid, this is outer membrane and outer layer. This is inner layer. Okay. And these are green color are called phospholipids. The phospholipid bilayer are arranged so that the polar ends of the molecule. I had shown the outer layer is polar end of the molecule which has the phosphate and the glycerol portion of the phospholipid which is soluble in water and it forms the outermost layer. And while the non-polar ends, which is called fatty acid portion, the fatty acid portion of the phospholipid that are insoluble in water, that form the central of the membrane. So the non-polar ends are the central of the membrane. which is the fatty acid portion, and it is insoluble in water. So, in the case of this phospholipid bilayer, I have shown outer membrane and inner membrane. That outer membrane and inner membrane are water soluble, and in between those, such as I will show it again, this layer is insoluble layer. This layer and this layer are soluble, water soluble. Next is the function of the cytoplasmic membrane. It is a selectively permeable membrane that determines what goes in and out of the organism. So it is capable of determining which molecule enter inside, which molecule go outside of the organism. All the cells must take in and retain all the various chemicals needed for the metabolism. Water, dissolved gases such as carbon dioxide and oxygen, and lipid-soluble molecule simply diffuse across the phospholipid bilayer. So these molecules diffuse across the phospholipid bilayer. The water-soluble ions generally pass through small pores, and the small pores are less than 0.8 nanometer in diameter within the membrane. All the other molecules require carrier molecules to transport them through the membrane. So it requires a carrier molecules for the transporting those molecules across the membrane. The materials move across the bacterial cytoplasmic membrane by different methods. How the materials move? It is mainly through passive diffusion, facilitated diffusion, and active transport. First, we can discuss about passive diffusion. The passive diffusion, it is the net movement of gases or small uncharged polar molecules across a phospholipid bilayer membrane from an area of higher concentration to an area of lower concentration. That is passive diffusion. Example of the gases that cross membranes by the passive diffusion includes nitrogen gas, oxygen gas, and carbon dioxide. And the examples of small polar molecules which move across the membrane phospholipid bilayer through the passive diffusion include ethanol, then water, and urea. These are the small polar molecules. So this is the cytoplasmic membrane, and this is the oxygen molecule, and this is the same picture showing the passive diffusion of the molecule. That is, here the concentration is higher, so it moves from the higher to lower concentration, and this is the net movement of the molecule, and this is an example for passive diffusion. All the molecules and atoms possess kinetic energy, the energy which is required for the motion. If the molecules or atoms are not evenly distributed on both sides of the membrane, if it is not equally distributed inside the bacterial cell as well as outside, so there will be a difference in the distribution of these atoms and molecules. So this difference, which in their concentration, which creates what? A concentration gradient will be created due to the difference in the concentration between inner as well as outer layer of the bacterial cell. And this concentration gradient represents a form of energy which is known as potential energy. It is also known as stored energy. The net movement of these particles will therefore be down their concentration gradient. So the net movement of those particles will be down the concentration gradient from the area of higher concentration to the area of lower concentration. So we can discuss about the different types of movements. First one is diffusion. In the case of the diffusion, it is powered by the potential energy. This is an example for the movement of the molecules across the cytoplasmic membrane. First example is diffusion, and this diffusion is powered by the potential energy. What do you mean by potential energy? The energy created due to the difference in the concentration gradient. So the diffusion is powered by the potential energy of a concentration gradient and does not require the expenditure of metabolic energy, and it doesn't require any expenditure of metabolic energy. Then coming is osmosis. Osmosis is the diffusion of the water across a membrane from an area of higher water concentration which have low solute concentration to the lower water concentration which has high solute concentration. This osmosis is also powered by what? The potential energy of a concentration gradient, and this osmosis also doesn't require the expenditure of metabolic energy. While the water molecules are small enough to pass between the phospholipids in the cytoplasmic membrane, their transport can be enhanced by water transporting transport proteins called aquaporins. So what do you mean by aquaporins? It is nothing but a transport proteins which enhance the transport of small water molecules. The small water molecules to pass between the phospholipid bilayer of the cytoplasmic membrane, their transport can be easily enhanced by the special type of transport proteins known as aquaporins. Those aquaporins form a channel. They form a channel that span the cytoplasmic membrane and transport water in and out of the cytoplasm. So, called channel proteins. So, to understand this osmosis that we have discussed in the previous slide, one must understand what is meant by a solution. So, what do you mean by solution? A solution consists of a solute dissolved in a solvent. In terms of osmosis, this solute refers to all the molecules or ions which are dissolved in the water, which are called solvent. When a solute such as sugar dissolves in a water, that is solvent, it forms a weak hydrogen bonds with the water molecule. What forms? Sugar forms a weak hydrogen bonds with the water molecules, while free unbound water molecules are small enough to pass through the membrane pores. So the water molecules bound to the solute are not. So what happens? Unbound water molecules. Some of this bound water molecules means those sugar molecules attached with the water molecule. Unbound is small enough to pass through the membrane pores. Therefore, higher the solute concentration, lower the concentration of free water molecules capable of passing through the membrane. Which means when the solute concentration is higher, so what happens? The lower the concentration of free water molecules. So the water molecules is very low because of the higher solute concentration. So it is not capable of passing through the membrane. This is the picture of osmosis. In the case of this osmosis, this is cytoplasmic membrane, and these are the bound water molecules, and these are the free water molecules. Bound water molecules, free water molecules.